You want to credit to your computer?
Yeah.
Oh, okay. Seems like that would be a good place.
Because cloud deletes it after so much
Okay.
time.
Okay, I'm gonna watch on to the other side. You might want to like unplug yourself right when you get up. I mean, good job.
We're gonna keep testing.
Yeah, I'm so sorry. Yeah, I know.
How are you?
Oh, maybe I should be in front of them. Just be a voice.
You can just be a voice.
Okay. Okay. It works for me. It's like, do we have to change seats again? So what time is it?
1:45.
So should we give people a minute
Yeah.
to come in? Those of you that are in the room, Amanda brought waters and cookies and oranges or little clementines if you want a bite. So, and walk around this way. You just throw it.
They are very tall.
That's right. Laura has a really good arm, so if you want a clementine, just raise your hand and she'll pass it to you. Food fight. Right, the only PhD defense that starts with a food fight. Yeah. Yeah. Just want to make sure. Okay, I think we can get started. It is my great pleasure to welcome you all for Amanda Benoit's thesis defense. I think that was my phone. I apologize. I'll shut it off. And I first of all like to thank her family and friends who have traveled to come and hear her defense. It's really an important day, and I'm going to get very emotional because we haven't been able to celebrate anything in person. And for me, this is just a thrill because Amanda has done such a wonderful thesis, and she deserves a lot of attention and praise and celebration. Amanda came to UT in 2016 with a master's degree, not a concurrent master's, but a master's degree from Guelph, where she worked with Christine Caruso on pollinator predators from a real ecological perspective. One of the things that I really love about Amanda's PhD thesis work is that it's very, I think it's really transformative. And people use that term a lot, but it really is, because she's taken the act of predation and looking at how it can affect the genetics of the plant population. And that's sort of leaping through pollinators into how the plants perform and what their fitness is really quite an amazing arc to have completed. So, and while Amanda's been here, she was highly decorated. She received a Rosemary Grant Award for her research from the Society for the Study of Evolution. She received a Graduate Research Award from the American Society of Naturalists. She's been hailed as a fabulous and wonderful teacher. She has actually taught classes herself as a graduate student, as an instructor of record, which is a very, not everyone gets to do that, let's just say. And so I'm really happy that you're all here to witness the final story and the pieces all coming together for Amanda's PhD. And I'd like to say that for two of our former lab members, Morgan and Amanda, who are in the audience, I know, and didn't get to have this in person, we'll just pretend it's all for all of us. Okay, and I'm going to turn the mic over to Amanda.
Yeah, thank you so much, Susan. Let me get this mic clipped on. Okay. So now I have a very short cord, so I'm like a little bit awkward and gonna try not to knock everything over. But I just want to say thank you so much for all coming. It's really wonderful to see all my friends and family and colleagues here in person. And I know there are a lot more of you out there in Zoom land. And with my current screen setup, I can't see who's here on Zoom, but I'm very happy that you can all be here for my talk. So I wanted to just start out with a bunch of thank yous because I haven't gotten to this point on my own. There have been so many of you that have helped me along this journey. So thank you so much to all my colleagues, especially my advisor, Susan Kalis, all my committee members, all the lab members who have helped me so much with this, especially Kristen, who's helped a lot with the lab work and the DNA extractions, my field assistant, Danny Coy, who spent hours and hours in the field with me, all the graduate students who have really made this journey such a wonderful experience, and all of my past mentors, especially my master's mentor, Christina Caruso, my undergrad mentor, Marcel Dorkin, and another undergrad mentor, Susan Letcher. Just so many wonderful people have helped me get to this point in my life. And of course, all of my family and friends and pets and wonderful animals in my life too. So my wonderful husband, Daniel Sixay, my parents, my friend Liz, all my sister-in-laws and nieces and nephews, and everyone who's here supporting me. And I just wanted to start off with a quote by Jane Goodall. So you cannot get through a single day without having an impact on the world around you. What you do makes a difference. And what you have to decide is what kind of a difference you want to make. So I think this is a really important message for all of us, especially in these sort of difficult world times when we're going through pandemics, climate change, wars. There's so much going on, and it's so difficult to feel like we can make a meaningful difference in the world. But I think it's important to remember that we can. And I think this is an interesting point that my research demonstrates too, that interactions, no matter how small scale, no matter how indirect, have the power to change the world, change populations, change communities. So I was thinking about how to start this, and I figured let's just go to my backyard. So here's my backyard. This is a scene that's probably familiar to everyone who's been on the East Coast of North America. It's this very green, very lush picture. And it might lead us to ask, why is the world green? So this is a question you might expect from a toddler, like, why is the sky blue? Why is the world green? But this is also a very ecologically important question. And it's one that spurred a lot of research over the past 60 years. And so on a very fundamental level, the world is green because of photosynthesis. Plants have chloroplasts with chlorophyll, and that gives them the extraordinary ability to turn energy from the sun, carbon dioxide from the air, and water into sugar and oxygen. So by creating sugar through this process, plants are the base of the food web and provide nourishment to all other living organisms, with a few minor exceptions if you're thinking about deep sea vents or something like that. But for the most part, all living life on Earth gets its food from plants. So plants are this really important base of the food web. And you've probably seen food web diagrams like this back in grade school, where we have, some nondescript grass at the bottom, and we really focus on all these charismatic animals up here. But these nondescript plants at the bottom are so important because they feed everything else. And the botanists in the room know that plants are so much more exciting than just the grass shown in this diagram. But the important point is that the plants produce all the food that feeds this first trophic level. So we have all these organisms that eat the grass, and then we have all these predators that in turn consume those primary consumers. And those predators have really important effects on the rest of the ecosystem by controlling the abundance and the behavior of those primary consumers. So in answer to the question, why is the world green? We've learned that it's really complicated. So we have these top-down effects of predators, but plants are not just passive players. They also have bottom-up effects. They have a lot of defense mechanisms. But the important part for our story today is that these predators can have profound cascading effects on plants via the herbivores, which we call trophic cascades. And this has been well recognized for a long time. There's over 2,000 published papers examining this. It's a very sort of well-established ecological concept. One of the most famous examples is this Yellowstone example, where we expatriated wolves. We hunted them out of the park in 1926. And after the wolves were gone, all the elk started coming down into the river valleys. You can see the effects. They ate all the vegetation, and then that changed the water temperature by decreasing the amount of shade. It decreased the bird habitat. So we had fish leaving or dying off. We had birds leaving. We had this whole community-wide change. And then we reintroduced the wolves to the park, and we saw that change reverse. So we saw all these plants start growing back in along the riverbanks. That created bird habitat. It brought the beavers back in because now there were saplings again. It shaded the water, so the fish came back. So we had these profound ecosystem changes because of predators. But predators eat more than just herbivores. So here's sort of a more nuanced version of that earlier food web I showed. So we have our plants as the foundation of the food web, but we also have this large array of primary consumers that are not all herbivores. So over on this side, we have the herbivores that have a negative effect on the plants, but we also have primary consumers that might have no effect on plants. And we have primary consumers that are mutualistic or have a beneficial effect on plants. So we have things like pollinators and seed dispersers that get their nutrition from plants, but they benefit the plants in the process. And then we have predators that consume all of these. And also they interact with one another through competition. So predators, by eating this whole wide array of primary consumers, can have different effects on plants. But so far, our research has really only focused on this side of this interaction, looking at predator effects through herbivores. So I got really interested in looking at, but what about the pollinators? How do predators affect plants through their pollinators? And for a long time, people thought predators don't really eat pollinators. Bees have stingers, maybe just nothing really eats them. But this isn't true. Lots and lots of things eat pollinators. So here are just a few examples. This is from my master's research. So ambush bugs will eat bumblebees and other pollinators. We have dragonflies. We have praying mantises that will even take out hummingbirds. We have spiders, lizards, all kinds of birds, including the genus called bee eaters, which obviously they eat bees. We have wasps, which are becoming invasive in a lot of areas. So why does this matter? Why does it matter if things eat pollinators? Well, plant-pollinator interactions are some of the most ecologically and evolutionarily important category of mutualistic interactions. So plants are literally rooted to the ground. They can't move around in search of a mate. So they've evolved this relationship with animal pollinators to move their pollen around and allow for outcross reproduction. Over 80% of flowering plants rely on animal pollinators. And so those pollinators have direct consequences for how those plants are able to mate, for their gene flow, their population genetics, their mating system, and their genetic variability, as well as their selection, because those pollinators have a direct effect on the fitness of those organisms and the relationship between the fitness and traits. So plant pollinators are extraordinarily important, not only ecologically and evolutionarily, but for all the food we eat. So economically important. We would not have a lot of our fruits and vegetables without animal pollination. And also, of course, pollinators are declining. So this is a conservation concern as well. So for my dissertation, I have 4 chapters. The first chapter is a review that really examines our current understanding of how predators, pollinators, and plants interact with one another, and makes some predictions and hypotheses for future directions. In my next 2 chapters, I went to the field, conducted some field experiments and observations, and also some greenhouse experiments, where I really looked at how predators affect plant selection and what the evolutionary consequences of those interactions may be, as well as looking at how the predators are really affecting the behavior of the pollinators and whether or not the pollinators are effectively pollinating, or if they're maybe just taking resources without being as effective pollinators. And then I look at the consequences for plant mating systems and inbreeding depression. So this review paper is out in annual reviews if you want to take a look. It's called Predator Effects on Plant Pollinator Interactions, Plant Reproduction, Mating Systems, and Evolution. So for this review paper, I did a literature survey to look at what do we know about predator-pollinator interactions. So I looked through the literature and found 62 different papers that explored interactions between predators and pollinators. And this is a summary of sort of who's involved. So I found a bunch of pairings of predators at the top and pollinators at the bottom. And the size of these boxes are representative of the proportion of papers that look at each of these types of organisms. So you can see we have a lot of studies on spiders. We have some studies on swallows. We have some on raptors and other predatory birds, some true bugs. I found it surprising there are actually 7 papers looking at frogs that eat pollinators. We also have wasps, flies, mantids, lizards, dragonflies. And then we're looking at who they eat. And a lot of these are social bees. So we have our honeybees, our bumblebees, other types of bees. We have some flies, we have moths and butterflies, and hummingbirds. And so now this isn't a complete picture, because this is really an area that's been not very well studied. So this is just a snapshot of what studies are available, what's out there, but this is not necessarily representative of all the pollinators and all the predators. I'm sure if we look harder, we'll find a lot more predators that eat a much wider array of pollinators. But even though it's pretty understudied, we do have evidence that there are lots of different predators that eat lots of different pollinators. And we also have some evidence about how they affect pollinators. So there are 2 main ways that predators can affect pollinators. So predators at the top here in red can either have trait-mediated effects or density-mediated effects. And that's just a complicated way of saying that they can change the behavior of pollinators, and they can change the abundance of pollinators. So some ways that they change the behavior are by decreasing foraging speed. So sometimes pollinators, in response to risk, will just slow down their foraging. They'll spend longer examining the plants before they land, or they'll just decrease their foraging overall. If it's a dangerous world out there, maybe I'll just stay in my house, like we've all been doing with the pandemic. And they can also share information, which is really interesting. So my sister-in-law here has bees. She's probably seen them doing their little waggle dance. And so honeybees can actually communicate information about the danger of a certain floral patch through the waggle dance they do. And other social bees like bumblebees will be more likely to land on a plant with another bumblebee if it's in a risky environment. So they can share that social information. And predators can also have a really significant effect on the population of pollinators. So for a long time, people thought, well, you know, predators are fairly rare, pollinators are abundant, so it's probably not a big deal. But we have some demographic studies and more recent evidence that shows that even a low abundance of predators with a relatively low prey capture rate. So if we say we have spiders on 2% of plants, which is a very common abundance measure, it can decrease bumblebee queen production by 11% over one season. So that's a pretty large effect on the population size of those bumblebees. So to recap, we have our plants. We know that they provide nectar and pollen to their pollinators. In return, those pollinators provide pollination services. So they move the pollen from the anthers of one plant to the stigma of another plant, providing for outcross reproduction. But as Laura will tell us, not always. So not all floral visitors are necessarily effective pollinators. And that's sort of a piece that's been missing in the current research. People tend to assume if you're an insect visiting a flower, you're a pollinator. But that's not necessarily the case. To be a pollinator, you need to actually be collecting that pollen and moving it to a plant of the same species and depositing it on the stigma. So some insects just come, they take resources, but they provide no service in return. And then we can add in the predator, which of course is getting food from the prey item, so from the pollinator, and in return is causing death and fear for the pollinator. So they're killing them and scaring them. And the plant is providing a hunting site for the predator. But is the predator having direct effects on the plant? We're not really sure. That's not something that there's much published data on, whether that predator is directly affecting the plant. But we do know something about the indirect effects. So there have been some studies that show that a predator on a plant will affect visitation frequency. So usually it decreases the number of pollinators that visit that plant, but sometimes they have different effects. So there are some spiders that can alter the reflectance of UV light and actually increase the number of pollinators that arrive at that plant. We know that they affect visit duration. So usually they shorten the amount of time that a pollinator spends on a plant. Usually the pollinator will land, go, ah, there's a predator, and take off and leave. So they stay for a shorter amount of time. And we know that that can sometimes affect plant reproduction. So there are some published studies out there that show plants with a predator on them receive fewer visits, shorter visits, and then they produce fewer seeds because they had less pollen received by those stigmas. Some current gaps in our understanding is whether or not these predators can indirectly affect selection on plant traits. So this is something I looked at in my master's, and I also looked at in a different system during my PhD. And really digging into this question of how they're impacting visitor behavior and how that relates to the effectiveness of those visitors as pollinators. So whether or not they're touching those reproductive structures so that they can pick up and deposit pollen, and how that affects the pollination biology of the plant. And then how that impacts population genetics, plant mating systems, and the expression of inbreeding depression. So I'm going to start by talking about my selection work. So just to recap, because I have a lot of family and friends in the audience. So natural selection can be easily sort of defined as the difference in survival and reproduction of organisms in relation to their phenotypic traits. So basically, if we have organisms with different traits, so if we look at everyone in this room, we all have different hair color. If hair color were somehow related to whether or not we survived and how many kids we had, then there would be selection on hair color. So if we think about how predators affect sort of the fitness and the traits of plants, I'm going to focus in on just sit and wait predators, which are predators that hunt directly from the plant. They sit on the flowers because flowers have reliable arrival of potential prey, right? Flowers attract a lot of insects. Predators eat insects. So those predators can sit on the flowers and wait for their prey to arrive and ambush them when they do. So we know that by sitting on a plant, predators make that plant risky to visit. So they're going to decrease visitation to that plant. What we don't know, what hasn't been studied, what hasn't been published on, is whether those predators are randomly distributed across plants or whether they're really associated with different plant traits. So if you think about how predators decide where to hunt, they're not making active decisions. They're not thinking it through logically, but they're just haphazardly choosing a place. They wait for some prey to arrive. If no prey arrive, they move to another place. So they have this process of trial and error. And by this trial and error process, they find hunting sites with lots of prey arriving, and then they stay there and hunt from there. And we also know that pollinator arrival is not random across plants. Pollinators can have trait preferences. So pollinators are collecting food. They're going to want to visit the plants that give them the most resources for the least time. So they're going to be looking for plants with lots of pollen, lots of nectar, bigger, showier flowers that they can see from further away, sometimes taller flowers that are above the other vegetation. So there's a bunch of traits that pollinators can be attracted to. And it seems like a logical prediction that predators may end up on plants with those traits because those plants have higher prey arrival. So if predators are associated with certain plant traits and decrease visitation to those traits and decrease the fitness of those plants, then they could affect selection. So we measure selection by regressing the standardized floral trait on the x-axis against the plant's relative fitness on the y-axis. So if we have a bunch of plants that range in, let's say, something like flower size that affects the attraction of insects, if all the plants across that range of flower size have the same relative fitness, then it'll look like this red line with no selection, because all the plants that those different trait sizes have roughly equivalent fitness. However, if the plants with larger flowers have higher fitness, then we would expect this black line, so that would be positive selection. And if the plants with larger flowers have lower fitness, then we would expect this blue line. And so we can also calculate selection gradients where we do multiple regression with multiple traits in that model at once. And then we get what we call a beta or selection gradient. So that is what I just described in this figure, the positive line, the horizontal line, or the negative line, but it's going to be in numeric form instead of a line on a graph. So that's what I'm going to be showing you in a little bit. So to see if predators can affect selection, I went to the field. So I went to UC Davis's McLaughlin Reserve, which is here in California. And I studied the yellow Mariposa lily, or Calcortus luteus, which is a plant in the lily family. And the distribution of this plant is shown by these blue dots. So it's endemic to California, but distributed around the Central Valley. And this is a plant that is interesting because it often produces only one flower, sometimes up to 6, but usually in the population where I was, each plant produces a single flower in a year, and that flower is only open for 3 days. And then the plants go dormant for the summer. In the fall, when it begins raining, they grow again and flower the next summer or next spring. And these are also very long-lived plants, so they can live up to 40 years in the field. So to study natural selection in the field, this is the field where I worked, and you can see the flowers in the field. So we went through the field and found plants that had crab spiders naturally present, and then selected a partner plant with no crab spiders present. So we had paired plants. We had 119 paired plants in 2017 and 150 in 2018. And we checked these plants daily throughout the flowering season to make sure crab spiders were present and absent. We measured floral traits, and then we collected fruits and counted seeds as a fitness component. So the first questions I wanted to answer in this population are, how abundant are the crab spiders? And are they distributed across those plant traits randomly, or are they associated with certain plant traits? So we found that crab spiders were quite abundant in this population. We found them on 7 to 9% of flowers, which is quite high. In the East Coast, common estimates are 1 to 2% of flowers have crab spiders. But some studies in the Alps have found spiders on upwards of 26% of plants. So it can be a pretty big range. It can really vary both spatially and temporally. And so some of the plant traits we looked at were floral depth, so sort of the height of the flower cup, flower width, which I'm just going to start referring to as flower size because it's sort of the size of that flower, and then flower height from the ground, so how tall that flowering stem is. And we found that in 2017, crab spiders were hunting from plants with wider flowers. So if we look at this sort of flower diameter, The plants that naturally were colonized by crab spiders were about 4 millimeters larger than the plants that were not occupied by crab spiders, and that was a statistically significant difference. However, in 2018, there was no difference in flower width, so the widths were roughly the same, and none of the other traits differed. So then we looked at selection. So remember I said we can have positive selection, no selection, or negative selection, and those are our betas. So here we have betas on our x-axis. So if they're overlapping with 0, that is this horizontal line or no selection. And if it's a positive beta value, that's positive selection. So as the trait values get larger, fitness increases. So you can see in 2017, there was no selection on plant height. There was no selection on flowering date, but flowering size, there is selection. And if you look at the yellow versus the blue, the yellow is the strength of selection on plants with no crab spiders, and the blue is the strength of selection on plants with crab spiders present. And so you can see that when there are no crab spiders, there's significant positive selection for increased flower size. So the plants with larger flowers had higher fitness, but when the crab spiders were present, there's no selection on flower size. So all the plants with the whole range of flower sizes had roughly equivalent fitness. There was no relationship between flower size and seed set. And then for both plants with and without crab spiders, we have selection for more flowers, which is not surprising. If you produce more flowers, you can produce more seeds. In 2018, selection looked a little bit different. So in 2018, we found that both plants with and without crab spiders had higher fitness if they were taller. So if they had the flower higher from the ground, more visible over the grass and other vegetation, they had higher fitness. We found that there was selection for earlier flowering dates. So that negative beta means that the ones that flowered earlier in the year had higher fitness than the ones that flowered later in the year. And this is not significantly different between plants with and without crab spiders. So they look like they're not overlapping, but statistically in our ANCOVA, there's no difference between those two. And interestingly, in 2018, there's no selection on flower size in either group. And again, we have selection for more flowers. So this is interesting because we can see what crab spiders are doing to the strength of selection on these plants when they're naturally colonizing these plants. But doing this sort of observational study also has its drawbacks because we can't necessarily attribute changes in selection to crab spiders. There might be other unmeasured factors that could be agents of selection. So to be able to really say that crab spiders are the agents of selection, we need to do an experiment. So that's what we did. We did a large experiment where we marked plants before they flowered. We randomly assigned them to crab spider present and absent treatments. And then we caught crab spiders in sweep netting and put them onto the plants that were assigned to the crab spider present treatment. So we set up 14 different 60-meter long transects. We tagged over 700 plants. There were 11 different treatment combinations. I'm not going to talk about all of those in this talk because that would be way too long. But I'm just going to talk about these two treatment combinations. We had plants that were open pollinated, so we did not manipulate their pollination treatment, but we did manipulate whether crab spiders were absent or present. And when we did that and measured selection, we found some interesting results. So when we manipulated crab spider presence, we found that there was a significant difference in selection on plant height when crab spiders were present compared to absent. So when crab spiders were added to plants, there was significantly stronger selection for taller plants than when crab spiders were not present. Similar to when we did not manipulate crab spiders, there's selection for earlier flowering. So that seemingly has nothing to do with the presence absence of crab spiders, but just earlier flowers did better. And again, we find this significant difference in flower size. So it's interesting because we found this in the plants in 2017 that were naturally colonized. In 2018, the plants that were naturally colonized did not have this difference. But in 2018, when we manipulated the presence of the crab spiders, again, we see that the crab spiders are changing the strength of selection on flower size. So plants with crab spiders present have no fitness difference, no matter what their size. But when crab spiders are absent, larger flowers have higher fitness. And this selection for flower number sort of decreases. We have not as strong selection on flower number when we manipulate the crab spiders. So next, we wanted to just look at the raw sort of seed set data. So we were using seed set as our fitness component for the selection analysis, but I also was just curious how spiders are affecting seed set when we just look at the raw data without relativizing it. And interestingly, we found that crab spiders increase seed set. So that was contrary to our predictions. We thought that, having a spider should decrease pollinator visitation and that should decrease seed set. But in both 2017 and 2018, we find that the plants that were naturally colonized by the crab spiders had significantly higher seed set than the plants with no crab spiders. And even when we experimentally manipulated the presence of crab spiders, we found that the plants that we put the crab spiders on trended towards higher seed set. So this is not quite significant statistically, but there is a trend towards higher seed set in the plants with those crab spiders. So to recap, we've now found some evidence that crab spiders are able to significantly alter the strength of selection on plant traits, especially flower size, and that they do affect the fitness of plants, but surprisingly, they increase seed set rather than decreased it. So next, we're going to look at visitor behavior. Maybe this will shed some light on why seed set is increasing. So again, we use the same study system looking at interactions between Calchordus luteus, crab spiders, and its pollinators. So this plant was reported to have a generalist pollinator system. It's a nice open cup flower that really any insect can visit. It's not a special shape that they need to fit inside. It's open, it's large, it produces nectar, it produces pollen. So it's visited by a wide range of insects. So we decided to do some pollinator observations on paired plants. So I found pairs of plants that were in roughly the same location in the same flowering stage. And then I would flip a coin to decide which one got a crab spider and which one did not. So then we put a crab spider on one of those flowers, and then my field assistant and I would watch each of the flowers for 15 minutes and observe how many pollinators or visitors came to the flower, how long they stayed, whether or not they made stigma and anther contact. And we did our best to identify them, and we photographed a bunch of them and identified them to genus based on the photographs. So from this, we found that the presence of a crab spider, which was experimentally manipulated, so the plant that we put the crab spider on, received 61% fewer visits per 15 minutes. So in that 15-minute observation period, there were 61% fewer insects that came to that plant. Of the insects that did visit the plant, they stayed for 48% less time. So there was a much shorter visit. And then we looked at whether the insects were touching the stigma and the anther. And so for those of you who never took a botany class, this is the stigma in the middle. That's what receives the pollen, and the pollen tubes grow down through to where the ovules are, and it fertilizes the ovules at the base and creates the seeds. And around the outside, we have the anthers that produce the pollen. So to properly pollinate, an insect will need to pick up pollen from one plant, fly to another plant, and deposit that pollen on the stigma to provide for outcross pollination. However, these plants are also self-compatible. So if pollen gets from these anthers to this stigma, it can produce seeds with itself. So the same plant can act as both the mother and the father. So looking at whether insects touched the anther and the stigma gives us an idea of how effective they are. And we found that when crab spiders were present, the number of stigma contacts, so the amount of stigma contact that the insects made was 77% lower. So 77% fewer of the insects actually ever touched the stigma. And anther contact was reduced by 80%. So what happened is often the pollinators would just be on the outside of these petals collecting nectar and never touch any of the reproductive structures. And I thought that I could look at different taxa of insect visitors and look at how crab spiders affected the different taxa and see if, maybe butterflies responded differently than bees, or if bumblebees responded differently than solitary bees. But it turns out that 97% of the insects I observed during these 45 hours of observation were in the same family. So they were halictid bees in 2 genera. So really, I didn't have the diversity of visitors to look at how crab spiders impact those different taxa in different ways. But given that amount of reduction in visitation and visit duration, I wanted to see if plant reproduction became pollen limited. So pollen limitation is when the seed set of the plant is limited by the amount of pollen that the plant receives. And this can be measured by comparing the seed set of plants that are just open, naturally pollinated to the seed set of plants that you give extra pollen to. So we have plants that are hand pollinated with supplemental pollen. So those plants on this bottom here have all the pollen they could possibly ever need. So we just put lots of pollen on them. The ones on the top just are receiving natural insect visitation pollination. And then we have plants with no crab spiders, and then plants that we placed crab spiders on. And this will allow us to calculate the magnitude of pollen limitation for plants without crab spiders and with crab spiders, and then to compare those to see if crab spiders are changing the magnitude of pollen limitation. But surprisingly, we found that seed set is not pollen limited. So pollen is not a factor that is limiting the reproduction of these plants. So here we have the plants with supplemental pollen. They produce about 29 seeds. And the ones that were open pollinated produced roughly the same number of seeds. It's not statistically different. And so it also, we did not find a statistically significant difference between whether the crab spiders were present or absent on those open pollinated plants. But it does sort of trend in the direction that we saw. So the crab spider addition plants are in red. The crab spider absent plants are in this yellow. And so it's sort of trending in that direction of when they're open pollinated, the ones with spiders are producing slightly more seeds than the ones without spiders. So one likely reason that these plants are not pollen limited is that it gets very dry during their flowering season. And in a related species in the same genus, there was a study that shows that reproduction is very water limited. So if you add supplemental water, these plants are able to produce many more seeds. And this is something we saw in the greenhouse experiment that I'm going to talk about in a minute too, is that the plants that we grew in the greenhouse and were watering every 3 days produced way more seeds than the plants in the field. So it may just be that plants are more water limited than pollen limited, so they just can't produce more seeds than they have water to produce. Okay, so to summarize, these predators are indirectly affecting selection, fitness. They're decreasing the visitation of pollinators, decreasing the duration of visits, and decreasing stigma and anther contact. So next, we wanted to look at this question of population genetics, mating systems, and inbreeding depression. And this brings us back to that question of why are the plants with crab spiders present producing more seeds despite lower visitation? And so there are several possible explanations for this. One possible explanation is that the crab spiders, when they're naturally choosing the plants, are just choosing higher quality plants. So they might be choosing plants to hunt from that just naturally have higher fitness. But we also saw the trend towards higher seed set when we manipulated the crab spider presence absence experimentally. And so that was only a trend, but still it suggests that there might be something more going on there. It could be that the predators are decreasing herbivory in addition to decreasing pollinator visitation. So there are herbivores like caterpillars that will come in and eat the flowers. There are also seed predators that will lay their eggs on the immature fruit, and then those eggs will hatch and the larva will consume some of the seeds as the fruit is developing. But if we exclude all the plants that had herbivory damage and just look at the seed set of plants that had no herbivory damage, we still see this difference in seed set. So that's not the only explanation. Another thing that's possible is decreased larceny. So if the plants are not pollen limited, then having extra pollinators coming and taking nectar, taking pollen, they're taking resources without providing any additional benefit because the plants have enough pollen. So decreasing the number of visits may decrease the amount of resources that the plant is giving to pollinators, which then may increase the amount of energy it has left to produce seeds. And lastly, the one that I'm going to focus on is that crab spiders may also be mediating self-pollination within the flowers. So these spiders, as they crawl around within the flowers hunting, may be delivering pollen from the anthers to the stigma. And that could be causing that increase in seed set and that sort of lack of relationship between flower size and seed set. So if crab spiders are mediating self-pollination and increasing rates of selfing, so selfing is when the plant is acting as both the mother and the father to those seeds rather than outcrossing, where you have two separate parents. This may increase the expression of inbreeding depression. So every organism or every diploid organism has two copies of each gene, and different versions of genes are called alleles. And so if you have the same allele at both your copies of it, so sorry, you get it better. So if your two copies of a gene are the same allele, you're homozygous. And if you have two different alleles, you're heterozygous. And if you're more inbred, you're more likely to be homozygous because you're more likely to have gotten the same allele because you have the same individual as your mother and father. So there's only two allele options rather than four allele options if you had four parents. And so if you're homozygous and those alleles are deleterious recessive alleles, then you may suffer from inbreeding depression. So a lot of recessive alleles may have a consequence for fitness. They may be slightly detrimental, but it's hard for natural selection to weed them out of the population because if an individual is heterozygous, there's no fitness consequence. So those alleles are maintained in the population. And then if you have inbreeding, you can have homozygosity and those deleterious recessive alleles are expressed and they decrease your fitness. So inbreeding can lead to decreased fitness, but whether or not it does will depend on how many deleterious recessive alleles are present in the population. So we wanted to determine whether the offspring of plants with crab spiders present are more inbred than the offspring of plants without crab spiders present. So we started by growing plants in the greenhouse, which we'll talk about in a second. And then we collected DNA from 33 maternal families and 5 individuals per family. So 326 plants that we collected DNA from. And these are plants that are the offspring of the field plants that had crab spiders present and absent. We extracted their DNA using Quiagen DNA-easy mini kits. And then we sent that DNA to the University of Minnesota Genomics Core, where they did genotype by sequencing or GBS analysis to get us the sequence data for all those individuals. I then sent that sequence data to my collaborator, Haran Zhu at the University of Toronto, and he cleaned up that sequence data using the STAX pipeline. And then we were able to look at the inbreeding coefficient for those individuals. So of the plants in the families that were from maternal plants without crab spiders, we see a slightly negative inbreeding coefficient, which indicates that those plants actually have a slightly higher heterozygosity rate than expected. So those are slightly more heterozygous than we would expect by random mating in that population in the Hardy-Weinberg equation. Whereas plants from mothers with crab spiders present have a slightly positive inbreeding coefficient, although it's not significantly different from 0. And these are not quite significantly different from one another. So this is just a trend. So we can't make too much of it, but it does seem like there's slightly more homozygosity in the offspring of the plants from maternal plants with crab spiders present. And one caveat to this is that the DNA was extracted from these plants 3 years after they were grown. And during that time period from germination to 3 years old, there's some mortality. So there were some seeds that never germinated, and then there were some that germinated but died. And so if there is inbreeding depression, the individuals that died may have been the ones that were more inbred. So we're actually seeing less inbreeding because these plants are the ones that survived and made it. We might have seen more if we had genotyped the seeds before germination. So I mentioned a greenhouse experiment. I did a very large greenhouse experiment with seeds collected from 4 different maternal plant treatments. So in the field, I had plants that I outcrossed. So for these plants, I cut off all of their anthers. I emasculated them so that they could not self-pollinate. They did not have any pollen. So all of the seeds had to be produced by outcross pollen. So I know these seeds were outcross pollinated. And then I also self-pollinated some plants. So for these ones, I put mesh bags over the flowers so that they could not receive outcross pollen. And then I hand pollinated them with their own pollen. So I know that these seeds were self-pollinated. And then I had maternal plants that had no crab spiders and open pollination, and maternal plants with crab spiders present and open pollination. So if there is inbreeding in this population, and if crab spiders are facilitating self-pollination and increasing the amount of selfing, then we would expect the self-pollination rate and inbreeding depression to be expressed from least to most, from left to right. So we would expect the fitness of those offspring to look something like this, with the outcrossed having the highest fitness, the self plants having the lowest fitness. And if crab spiders are causing more selfing, then we would expect the offspring of plants with crab spiders to be more similar to the self, and the offspring of plants without crab spiders to be more similar to the outcrossed. So I planted over 2,200 seeds 4 years ago in 2018, and I've been tracking the fitness metrics of those seeds for the past 4 years. So I looked at what percentage of those seeds germinated, how they've grown, which ones have survived, and most of them flowered in their 3rd year of growth, so last year. So I measured floral traits and then how many seeds each of those plants produced. So here's just a survival metric over those 3 years. So this is survival as of January 2021. And so we have our outcross plants here with the highest survival. We have the offspring of maternal plants without crab spiders that have basically exactly the same survival rate as the outcross plants. We have our selfed seeds, which have the lowest survival rate over those 3 years. And then we have the offspring of the maternal plants with crab spiders with an intermediate survival rate. And so the maternal plants with crab spider offspring are not significantly different from the selfed, but they are also not significantly different from the other two groups. But the outcrossed and the maternal plants without crab spiders are significantly higher than the selfed. So I'm going to go through some comparisons of the outcrossed and self seeds and their fitness. So if the self seeds have lower fitness, this is evidence of inbreeding depression in this population. And we do find evidence of this. So the selfed plants are significantly less fit than the plants that were produced via outcrossing. So we found 2% lower germination, which was not significant statistically, but they had 14% lower survival, which is significant, and also 27% fewer plants flowered in their 3rd year compared to the ones that were outcrossed. And of those that flowered, they produced 62% fewer seeds. So here you can see the seed set of the outcrossed plants. And you can also see that this is much higher than it was in the field. So as I said, these are plants that are being watered every 3 days. They're being fertilized. They have no competition. They're in a very cushy greenhouse environment where we're doing everything that they could ask for. So that's really affecting the fitness and survival of these plants and might be decreasing the apparent or the observed level of inbreeding depression, because if they were in those harsher environments, we may have seen even greater differences in fitness if inbreeding depression reduces their ability to cope with harsh environments. So next, I'm comparing the offspring of maternal plants with crab spiders present to the offspring of plants with maternal plants with crab spiders absent. So I found 4% lower germination. However, that was not significant. Also 2% lower survival. That was not significant. Actually 4% more flowered in year 3. So that's sort of an opposite direction than what we would predict. And seed set did not differ. So this is sort of mixed evidence. So we definitely know that the outcross plants are more fit than the selfed plants. It's a little bit less clear what's going on with the offspring of maternal plants with crab spiders present and absent. And like I said, that may have something to do with the fact that we're growing them in really cushy greenhouse environments that may be masking inbreeding depression. It may also have to do with, not all of the offspring would have been selfed. Some of them would have been outcrossed, some of them would have been selfed. And it's possible that the ones that didn't germinate and then died were the ones that were selfed. So there's some interesting population genetic stuff going on that we are still digging into. And I guess one important thing to point out is that the genetic data that just came back from my collaborator 2 weeks ago. So I got those results back the day that I was sending my dissertation to my committee. And I would like to dig into that data in a much more robust way. So I want to look at the genetic distance between each individual within a family and then get family level estimates of genetic distance and compare those. I think that'll be a much more robust way of looking at that data than just looking at the inbreeding coefficient. So this is still very much exciting frontier where we're just starting to really dig into it. But I think it's really exciting to see that there are maybe some population level genetic consequences of having a spider in flowers. So to come back to our food web at the beginning. So like I said, we have lots of evidence for this side of the web that predators affect plants through their herbivores. And I think one of the reasons that this is so much more obvious is that herbivores have this really immediate impact on plants, right? If a deer comes along and eats a trillium, that trillium is gone, it's eaten, it's an immediate effect on fitness and biomass. Whereas if a bee comes along and pollinates that trillium, that's going to take longer. It takes a year for those seeds to then get dispersed and to germinate and then to see the fitness effects of those offspring. So it's a much more delayed process when you're looking at mutualism versus herbivores. So I think it's very possible that predators are also having really profound effects through mutualists, but they're just a lot harder to measure and to see because the timescales are different. And another thing to point out is the fitness effects that we saw in our greenhouse study between those self and outcross plants took years to show up. So if you just look at the seed set of plants that were self-pollinated and plants that were outcross-pollinated, they produce the same number of seeds. And that's as far as researchers sometimes go. They go, okay, self-outcross, they produce the same number of seeds, they have the same fitness. But it's really important to consider the fitness of those offspring. Do those seeds have the same vitality? Are they going to be able to survive in the same way? And what we found when we grew them for 4 years in a greenhouse is no. The ones that were outcrossed had much higher survival, much higher growth. They produced more flowers, they produced more seeds. And the ones that were selfed had much lower fitness over those 4 years. But you had to grow them in a greenhouse for 4 years to see that. So it takes a lot longer to see those differences. So to come back to my quote, you cannot get through a single day without having an impact on the world around you. What you do makes a difference. You have to decide what kind of a difference you want to make. So I hope all of you decide to make a positive impact on the world. And to bring it back to my spiders, plants, and bees, no organism can exist without having an impact on the world around it. So interactions, whether direct or indirect, shape populations, communities, and the world. And I'd like to thank all the agencies that have provided funding for this work. And with that, I'm happy to take any questions.
So one thing, if I could just say. So this is both the exit seminar and the defense. So what we typically do is have the people who are not part of Amanda's committee ask questions now. And then when you're done asking the questions about her work, you leave the room and then the committee stays behind and will continue to discuss her results and ask questions of Amanda. So this is your turn if you're in the audience or the physical audience or on Zoom to ask a question. But I'm not seeing if people are asking questions.
I can pull that up too. Yeah. Wait, I can't. With the laser pointer, I can't click. So maybe you should log in. Question?
I have a question. Well, actually, I have a question only if I'm allowed to ask a philosophical question.
Oh, goodness, yes, okay.
So the way that you bookended your seminar today, we're both taken up in like familiarized and philosophical jargon here. You were giving ontological perspectives on the information that the work that you're doing in ecology and evolutionary biology structures the way that we ought to see the world and the way that we ought to organize the world most productively, most interestingly, most novelly. Seeing the change in perspective that you're presenting here versus the perspective that you were gesturing at in your master's work, it seems like you've come to some interesting ontological conclusions of the work that you've been doing. I wonder if you had any thoughts about what those might be.
For those of you who don't know, Daniel's a philosopher. Yeah, so I guess I've come to really appreciate the importance of interactions and the effects of those interactions and the way that they structure the world more so than believing in individuality. You know, the community is more important than the individual. You know, we are all a sum of all of our interactions more so than we are an individual. So yeah, I guess my worldview has come to appreciate connections and interactions a lot more so than individuality.
That's what I thought, sorry, that's what I thought I saw in the shift, that your ontology is switched from individualistic ontology to a relational ontology.
Yeah. Erin?
I have a question for those humbly minded Joe Blows out there. In your research, and especially with how the climate is now, did you have any observations maybe what the average Joe can do to help the environment as a whole?
Oh, I love that question. Thank you, Erin. So for those of you on Zoom, the question is like, given climate change and all that, what can regular people do? And this is not something that my research goes into, but this is something that I've gotten very interested in sort of on the side. And those of you who attended Doug Talamay's talk here a few months ago, it's very much, that's the direction I'm going. So Doug Talamay is this eminent entomologist who, I think I gave you his book for Christmas. Anyways, so he has this idea that really to help conservation, we need this grassroots effort. He calls it the homegrown national park. Whereas if everyone were to convert half of their lawn to native plants, those native plants support the rest of the food web, like I was saying in my introduction. So instead of just having that simple grass ecosystem that feeds basically nothing, if we have native plants, those plants feed a wide variety of insects. Those insects then feed a wide variety of predators like birds and other fun organisms. So really we can sort of reverse a lot of the habitat loss and species decline we've seen just by focusing on planting native plants. And that will really make a huge difference and help restore a lot of these food webs.
Thank you.
Yes.
First off, very well done, and it's a very elegant research design, so nice work. I
Thank
have two questions. The first one is, do you think there are any environmental conditions that explain the differences you saw in 2017 and 2018, focusing on the observational study of pollinators that visited this flower?
Yeah, so that's something I'd like to dig a little bit more into. I think there was lower fitness overall in 2018. So I'd like to look back at the rainfall records and see if the temperature, I'm guessing it was probably hotter and drier. And that was affecting the fitness of the plants. Also, crab spiders were 2% more abundant in that year. So they were on 9% of plants rather than 7% of plants. I'm not sure how the abundance of pollinators differed in that year. But I think, the plants are very long-lived. Their abundance doesn't fluctuate too much year to year, but the insects and the spiders can fluctuate pretty significantly year to year in their populations and abundances. And then also herbivores can affect the plants pretty significantly, and rainfall, temperature.
And then second, you had me thinking a lot about like what would cause self-pollination, and it seems like a crab spider could do it moving around the flower, or other pollinators can
Yes.
do self-pollination.
Yes.
But I guess what I'm curious about is there any evidence in the literature or from your experience that crab spiders could actually be outcrossing plants and therefore be pollinators?
I have thought about that. I think it's probably not very likely, just because crab spiders are not super mobile. So they're pretty chunky and like they can't fly like insects can. So they have to literally crawl from plant to plant. And it's a pretty, if you look sort of into the field, let me get a field photo. The plants are, several meters apart, and they're in this sort of like rocky, dry terrain. So it's sort of hard for a crab spider to like crawl down from a plant, crawl across the ground, crawl up to another one. So they might do some very limited outcross pollination, but probably not a significant level. I guess the one way that they may be increasing outcross pollination, which I did not talk about in this talk, but which I write about in my dissertation, is that when they do capture an insect, they sometimes sort of run up to that top of the stigma and struggle with them there. And then as they're trying to subdue and kill them, like wipe the pollinator across the stigma. So they might just be really increasing deposition of pollen as a consequence of their predation activity.
So you don't have any evidence of ballooning
The little ones can balloon for sure, but once they're really big, they can't anymore. Oh yeah, for those of you who don't know, ballooning is super cool. The really small spiders just send up silk lines and can like float around, and they can travel miles. Like they can disperse really far just floating through the air. So that's how spiders disperse to new places.
I can't, I'm getting like.
Okay, let me try to pull up my chat questions.
Can you hear me, Amanda?
Okay, so Lucas has a question.
Oh, there you go.
You show that plants with higher height and larger flowers are correlated with the presence of spiders. I wonder how to differentiate this pattern reveals an effect on the spiders, on flowers, or just a preference of spiders for specific traits. So yes, so the spiders only had a preference for flower size, and that was only in 2017. There was no preference for height. And my guess is that the spiders are moving around until they find plants with high prey arrival and then staying there and hunting. And so likely pollinators are visiting larger flowers more frequently than smaller flowers, which is a trait preference that is pretty common among pollinators and other systems. Flower size is often associated with increased pollinator visitation. And so my guess is that the spiders are just following the pollinators, and the pollinators are going to the larger flowers. Let me see what. Okay. Is it possible that spiders also target nectar production levels? Any evidence for nectar variation and trade-offs with flower size? Oh, that's a good question. So I did not look at any nectar traits in the field in this system. However, there are some studies out there that suggest that crab spiders do drink nectar as a source of hydration. And if they're not able to capture insects, they can sustain themselves on nectar for some time. And also pollinators usually tend to visit plants with higher nectar production. So it's possible that they're both queuing in on nectar, and that nectar, you know, is sort of an unmeasured trait that may then correlate with flower size.
Great answer. Thanks. Nice presentation.
So I think I saw that one already. Yes. Okay. Any other, Jesse?
Yeah, so you talked about the prevalence of spiders being 7 to 9%, and I'm wondering, so is that over all of the flowers that you observe on a given year, or like what? You talked about how the flowers only last for 3 days, right?
Yes.
So you go out in the bee and you're foraging on a given day, you have a certain number of flowers out there. Does the prevalence of spiders on open flowers change throughout the season?
Oh, that's a good question. I have not looked at that, but anecdotally, I want to say yes. So anecdotally, when the flowers first start blooming, the spiders haven't sort of figured out that those are good hunting sites yet. So it takes a little bit into the flowering season before you start seeing lots of crab spiders on these plants. So the crab spiders can live for, you know, a number of months. And so they're switching, you know, throughout their lifespan from one species to another species as hunting sites. And it takes them some time to realize like, oh, you know, this species might have higher prey availability than this other species. And I'm anthropomorphizing. They're not actually logically thinking that. But they can also change color depending on what plant they're on, but that's sort of energetically expensive and can take a number of days to happen. So I think it takes maybe a week or two into the flowering season for them to turn yellow and red and to really start hunting from those plants. Yeah.
I have one more question, if that's okay.
Yes.
So when you're looking at the effects of predators on selection, you compare the treatment with only spiders without spiders, right? So selection in a artificial population that was devoid of spiders and one in an artificial population that all had spiders. And
Yes.
you know, they're being visited by the same set of pollinators. But I was just curious, given that the crab spiders had slightly higher plants with the crab spiders have slightly higher seed set, but tended to, like, you know, neutralize that slope on flower size. If you're thinking about an actual population with an actual prevalence of crab spiders, are you thinking it would just like weaken that slope a little bit, or do you think it would change it at all?
Yeah, so sort of thinking big picture, I think if you have high predator prevalence over a number of years and they're weakening selection on those traits that attract pollinators and just decreasing the pollinator population, predators could potentially play a role in shifts from mixed mating systems to self-pollination mating systems if they're making attracting pollinators less profitable and sort of decreasing the strength of selection on traits that attract pollinators. You may see a shift in selection towards more selfing traits where the plants just don't pump those resources into attracting pollinators and just focus on selfing, where then you get the transmission advantage of passing on all of your genes, but then you have the negative effects of inbreeding depression. But some highly selfing populations can purge those deleterious recessive alleles and just self. So that's one sort of thing that I talk about a little bit in my review paper, is that the predators, if they're prevalent enough over time, may contribute to the evolution of selfing mating systems. Yes.
I have a question. So did you pick the 3-day blooming lily because the prevalence of that's where the spiders will be? Or was that just an opportune environment for your experiment? Like what if there was another flower that bloomed for maybe 2 weeks?
Yeah, no, this is an interesting question. It's sort of always a roundabout way that everyone finds their study system. And so the only reason I wasn't necessarily going to go to California, but then I got a grant that gave me the money to go do the field work. And so I went for it. And I initially was going to go work on Collinsia, which is the genus that Susan works on and has a lot of background information on that genus. And it's also an interesting genus because it has repeated evolution from outcrossing to selfing. So I was sort of interested in this question of how predators may affect the evolution of selfing. And Susan had this lovely picture of a crab spider eating a pollinator on a Collinsia plant. So I submitted this grant to go look at Collinsia out in California. I got there, I looked at about 2,000 Collinsia heterophila plants and found 3 spiders. And so that clearly was not a viable system. So while I was there, I just started looking at other plants, and I found that these lilies were, you know, really abundant, and that they had these really high rates of crab spiders, and it just worked out as a really good system to answer the questions I was interested in.
Okay, so that was a lot of big words. Very well done. I'm just blown away by this research that you've done. I just want to make sure I understand because it's very interesting to me. So if, like, do the pollinators learn not to pollinate the flowers with the spiders on them?
Yeah, so there's some interesting lab studies on this where they actually have like bumblebees or honeybees in a lab, and they set out artificial flowers and they simulate artificial predator attacks. So basically they take a pair of tweezers and squeeze the bees when they visit some of the flowers to simulate forceps of a spider pinching them. And the bees can learn which flowers they get pinched on and avoid those flowers. Or if the artificial flowers that you pinch them on are all one color, the bees will learn to avoid that color and only visit the other flowers. There's even sort of, if you pinch them on more rewarding flowers, they can learn to forage on less rewarding flowers. or they just slow down and they start looking at the flowers for a long time before they land. And so yeah, I haven't done that research, but there is sort of a body of research out there looking at the bee learning and how there's no evidence that they innately recognize and avoid predators, but that through experience they can learn, which is important because body size may then play a role, because the bigger the bee, the more likely it is to survive an attack. So then it has the opportunity to learn. Whereas if it's a small bee, it may be killed on its first attack and then not have the opportunity to learn. And then that's also another sort of gap in the research, because we have some information about social bees and how they learn and how they respond. But there's really not much work that's been done on the sort of behavior and learning of other types of bees or just other taxa of pollinators. Mhm. Any more questions on Zoom? There's only 10 people left on Zoom. I don't know how many there were before.
Okay, well, thank you all for coming, and let's give Amanda.
Thank you so much, everyone.